Dear students, today we are going to study point double fertilization from your chapter Reproduction in lower and higher plants, its process and significance. So first let us try to understand what is fertilization. You have studied it in 10th standard. Fertilization. Fusion of male gamete with female gamete to form the diploid zygote is known as fertilization. So male gamete and female gamete. You know that haploid cells have n number of chromosomes. So n number of chromosomes from male gamete and n chromosomes from female gamete together make a diploid zygote. So this is fertilization, fusion of male gamete and female gamete. It makes a diploid zygote and this process is known as fertilization. Now let us see what is double fertilization. Double fertilization, fusion of one male gamete with egg and other male gamete with secondary nucleus is called double fertilization. Now see why it is double, because there are two fertilizations are there. One male gamete plus egg means here male gamete and female gamete, it forms zygote, and other is with male gamete and secondary nucleus. Now this type of fertilization, it is a unique character seen in angiospermic plants. Its discovery, it was discovered by scientist S.G. Nowsachin in 1898 in Lilam Fritillaria species. So he was studying this plant, and that time he studied double fertilization. Now before studying this process, we will revise some points, that is pollination. Pollination is transfer of pollen grains. So you have studied stamen, it is male reproductive organ. Stamen has a filament, connective and anther. Anther is bilobed. So this is anther. From this anther, pollen grains are released when it matures. So these are pollen grains. We have studied already this point. So from the anther, pollen grains are released. These pollen grains then get transfer on the stigma. Now stigma, you have seen this part in female reproductive organ, that is gynoecium, which consists of stigma, style and ovary. So from here pollen grains get transfer on the stigma. This is style, this is ovary. Inside ovary there is ovule, then all inner parts we will study afterwards. This is pollen grain. So this is pollination. Now when the pollen grain sits on the stigma, it is recognized by the stigma whether it is of right type, and if it is of right type, then after recognition its germination takes place. Germination of pollen grain takes place. Now at time of germination, what happens? Pollen grain comes here, and if it is of right type, it germinates and it produces a germinating tube, and this tube enters through the style into the ovary. Now we have seen that pollen grain, two cell stage of pollen grain we have studied. In that two cell stage, what we have studied, there are two types of cells. So these two types of cells are, one is vegetative cell, you can say it as a, this is generative cell, and this other is tube cell. Now during the formation of tube, what happens? This cell is there, generative cell. It forms two male gametes. This tube is formed. So inside all this cell cytoplasm and material is there, and this cell forms two male gametes. And tube nucleus is done. So this is tube nucleus. It means when the pollen grain sits here, gets attached to the stigma and starts with germination, it produces a pollen tube, and in that pollen tube the two male gametes also start moving. Now these male gametes are non-motile. They cannot reach the ovary. Inside the ovule they cannot reach on their own as they are non-motile. So the tube extends and the tube reaches till the female organ, that is till the ovule. Now entry of the pollen tube in the ovule is by 3 ways. And which are those 3 ways that we will see? You know this structure. Now here these are 2 synergids, 1 egg cell. So you can say this is egg cell 1 and 2, these are synergids, and these are antipodals. Now this is micropylar end, and here in the center, now these antipodals, 3 antipodals are there, and here 1 polar nucleus is there. So if the pollen tube, there are 3 ways in which the pollen tube enters. Pollen tube enters the ovule by any of the 3 ways. First, it may enter through micropyle. It is known as porogamy. Second, it may enter through integuments. It is known as mesogamy. And third, it may enter through chalazal end. It is known as chalazogamy. So these are the 3 ways through which the pollen tube enters. So if the pollen tube enters through the micropyle end, now we will consider as it is going through the micropyle end, means it is a porogamy. It enters into the ovule. Here it damages the synergids. Now here near the synergids, filiform apparatus are there. This filiform apparatus, irrespective of this entry, irrespective of the place of entry, it means it may enter through any place of the place of entry into ovule. A pollen tube always enters embryo sac near the egg apparatus. Meaning of this is, if it is enters through micropyle, if it enters through the integuments, or if it enters through the chalazal end, through any of these 3 sites, if it enters into the ovule. Finally it reaches the embryo sac near the egg apparatus. These 2 are egg apparatus. So through any way, finally it has to reach to the ovule, because there is a egg cell and the male gamete fuses with the egg cell. Here we have seen that the pollen tube enters. Now what happens here? When it reaches there, filiform apparatus of synergids attract the pollen tube towards egg apparatus. So here filiform apparatus of this synergids, it is attracting the pollen tube towards the female gamete. This is female egg cell. So it reaches till here. Now tip of the pollen tube absorbs water from one of synergids and bursts open to release two male gametes. And the tube nucleus degenerates. Means finally what happens here? Pollen tube has two male gametes and one tube nucleus. When it enters into the ovule, here synergids are there. One of the synergids provides water to this end and the tube bursts and it releases two male gametes are released. Now when it goes inside, two male gametes are released, process of fertilization starts, and let us see how is that process of fertilization. The tube nucleus degenerates, the released eggs bring out double fertilization. Now let us see which are the two steps of this fertilization. First, syngamy or first fertilization. So what happens here? Syngamy means one male gamete fuses with egg or ooze pair to form diploid zygote or ooze spore. So one male gamete fuses with the egg, means these are two synergids. This is egg cell. So one male gamete comes here. This is male gamete. So this is syngame. Egg cell and male gamete, it forms syngame. This is first step. Then second step is triple fusion or second fertilization. There are two fertilizations, so it is called as double fertilization. The first fertilization is with the egg cell and the male, and second is with the secondary nucleus and male gamete. So here what happens? Inside the ovule. Now here you have seen finally first this is first fertilization and second is here. There are two polar cells, then one male gamete fuses with it and it is triploid. Now this 1, 2 and 3, this is 3 and that is triple fusion. Two polar cells and that is the secondary nucleus and male gamete. So in this way the fusion takes place, second male gamete and secondary nucleus. This is triple fusion. Now this triple fusion, it is also known as PEN. PEN means the other male gamete fuses with diploid secondary nucleus to form the triploid primary nucleus. It is known as PEN, triploid primary endosperm nucleus. So this is called as triple fusion. What is triple fusion? Here the question may be asked, what is triple fusion? So triple fusion is between the secondary nucleus, diploid secondary nucleus plus primary one male gamete, that forms the triploid nucleus. So here completes the second fertilization. So in short, what we have studied, final figure of your double fertilization is two synergids, egg cell, male gamete. There in middle so the labels are First, these are 3 antipodals. This is chalaza. Then second male gamete. Then this is secondary nucleus. Now this second male gamete plus secondary nucleus, this is triple fusion. Then here male gamete, egg and damaged synergid. Now this male gamete and egg, it together makes a syngamy. Then from here our pollen tube is entering. This is pollen tube. And this is micropylon. So through micropyle the pollen tube is inserting here. It reaches the synergids. The filiform apparatus helps us to reach the egg. Then one male gamete fuses with the egg, it is called as syngamy. Other male gamete fuses with the secondary nucleus, it is called as triple fusion. And this is the total figure of double fertilization. So this is the process of double fertilization. Now its significance. What is the significance? And one more point here we have to study, that is meaning of siphonogamy. What is the meaning of siphonogamy? Siphonogamy. Fertilization process in which non-motile male gametes are transported up to female gametes through pollen tube.